Good morning. It's great to be here with you all. I'm a professor here in physics and bioengineering. I've been here for about 16 years, and over this time, my group has shifted its focus from really super resolution fluorescence microscopy to using those tools to understand what you see here in this picture, which are mitochondria and mitochondrial networks. So understanding the biophysics of those systems. And now on to the next, what we think is the next great thing, which is smart microscopy. And the overarching theme of all of this has been, driven by the motivation of myself and my group to exploit this virtuous cycle between technology development and discovery for cellular biophysics. So mitochondria are a relatively recent love of mine. As many people, you may, I was first exposed to mitochondria when I was first taking biology in high school. That was also the last time I really learned about mitochondria in a formal setting. But the thing that we learn about mitochondria is that they are the main source of the cellular currency for energy, ATP. And one of the things that we've come to be fascinated by since then is the fact that mitochondria, being given their evolutionary history as ancient endosymbionts, are actually something like a miniature cell within our cells. And so we've become really fascinated by the fact that they have their own life cycle within the cell. And that's really been more of our focus. And so we've been interested then in understanding their heterogeneous organization and dynamics. And our strategy for understanding that has been microscopy. And so since the early days of my lab, we've been developing these high precision light microscopy methods, super resolution, but also automation to be able to collect large enough data sets that we could start to suss out some of the patterns in their life cycle. And so for us, this has been a great set of tools, microscopy, automation, super resolution. And this is kind of a classical, closed loop control that you could picture having in a microscope. In a super resolution microscope, it's slightly different because of the way that you need to control the excitation and the emission of the fluorophores. But nevertheless, this whole idea of having a microscope that is imaging the sample, that's a sort of sensing component, and then in real time having an analyzer which figures out how you're doing in terms of the imaging, that performs a computation on that image, and then actuates the controller to then adapt the measurement. So that's the general idea. And we've combined this in my group with other types of optical engineering, in particular, expanding the fields of view of our microscopes by engineering flat laser illumination, or in the case of multifocal microscopy, which we've also built in our lab, creating uniform fields with multifocal laser illumination. So this has been kind of the technology that's been enabling for us. And how does this connect then to this problem of understanding mitochondria and their life cycle? So let's go a little bit there before we come back to smart microscopy. Really the advantage of being able to build these microscopes that are both automated and sort of higher throughput because they have larger fields of view is that we can watch the system and collect its spontaneous dynamics rather than having to perturb it to force it to do something when we're watching. And so we have access to the system without forcing it. And this is important because mitochondria are highly sensitive. They'll do different things if you force them than they will if they think you're not looking. And so here's a zoom in to a single cell. Here you can see the edge of the cell. And each of these little wormy structures is a mitochondrion. So you can see that mitochondria, they're moving in the cell, they're highly heterogeneous. And part of their life cycle is that they divide, but they also fuse back together. And so they communicate with each other, but they also divide to proliferate. And part of this the ways you can understand this is the fact that even though it was about 2 billion years ago that they became an energy source for what became the eukaryotic cell, they have maintained a minimal genome. So they have their own DNA copies. And so they need to replicate that DNA. And so that means that to make more mitochondria, you need to divide them. And so this division cycle is one of the things we're interested in. And I challenge you to look at this movie and find a division. It is really not simple. So there are people in my lab who have over the years developed, their neural networks to be able to detect mitochondrial divisions. And thanks to them, we have built up large enough data sets to use to train actual artificial neural networks to detect mitochondrial divisions. But this is a really challenging problem, as you could imagine. So, trying to catch these different parts of this life cycle in this very heterogeneous and dynamic system, where the DNA replication and division are highly asynchronous compared to our own cell cycle. So it's really a difficult problem of event detection that we're up against. So highly dynamic and heterogeneous, but also very sensitive to photo damage. And so here, we're imaging mitochondria. This is a sensor of mitochondrial membrane potential. And you can also see the endoplasmic reticulum in red. These two organelles communicate with each other via calcium signaling. And over the course of this movie, you can see that the mitochondria change their shape. They lose their internal structure. They lose their membrane potential, which is seen as a diminishment of the green fluorescence. And so all of these things are issues that we run up against when we're trying to capture the life cycle. And so nevertheless, researchers and biologists, extremely persistent. You take lots of short movies, you eventually collect enough data to study the different features of the system that you're interested in. But once you've done that for a while, you think, as a technologist, you think, can we do better? And we're always trying to push the technology to do better. And one of the things that we started to get really curious about, and this was, partially inspired by this really nice editorial by Jan Husken and Nico Scherf. Jan Husken is a great developer of light sheet microscopy, is this idea of smart microscopy. And I think the title of this editorial was a smart and gentle microscope. And the idea here is that you could build a microscope that adapts to the sample. And this is just a clip from that. Acquisition and processing must be tightly connected so that the microscope adapts to the sample, acquires only what is essential to the particular question, and leaves the rest untouched. So this sounds beautiful, right? And this is very much what we would like to be able to do, to leave the system relatively unperturbed and just get the information that we really need to answer our questions. And so part of this then is automation. And we think about a typical imaging workflow as being sort of this linear workflow where you have your system that you're studying, you put it on the microscope, you take images, you take those images, and perhaps now with modern tools, you use different forms of artificial neural networks to process, or machine learning to process this data. And then you output some kinds of analysis and you try to make sense of the data this way. And what we and others in this smart microscopy field are doing is to kind of close this loop and to turn this into an adaptive imaging workflow that incorporates the neural network into driving the microscope rather than simply using it at the end to analyze the data. So one of the things that I thought in the beginning is that, well, given that people are developing all of these artificial neural networks for segmentation and other tasks, perhaps what we could do is simply repurpose those same neural networks to drive our microscopes. It's not quite that simple. And closing this loop is not quite that simple. And event detection in dynamic systems is both a different challenge and offers different opportunities for detecting more subtle events, in my view. So we're working within an open microscopy framework with PyMM Core Plus as our control software. All right, so back to mitochondria. One of the things that we were really curious about mitochondria is, their life cycle, as I've mentioned. And so one of the patterns that we initially started to study is where and when do mitochondria divide? And so after lots of human effort, this is a montage of just about 20 mitochondria, and you can see that mitochondria have all these different lengths and sizes and branching morphologies. We had identified these division sites, and we did this, in fact, for hundreds of individual mitochondrial fissions. And we discovered that there are patterns in this division, but we could also take this data and indeed use it to train a neural network. And we did this not only through identifying where these sites were, but also by, we needed to define a ground truth. And this was an important element. What is the actual event score that we will assign to each of these sites? And I'm not going to go into the details of that here, but it turns out to be quite an important problem. So we trained a relatively classical architecture, a UNet architecture, with this kind of data. For this initial application that we did, we were using both the mitochondria, the shape of the mitochondria, that is the mitochondrial staining, and a protein which enriches at the sites of division. This protein doesn't only enrich at the sites of division, so it still is a relatively subtle problem. But you can see here these red spots, and those red spots correspond to DRP1, the protein responsible for dividing mitochondria. And here you can see that the neural network has given a higher score to this site than to this one. And so it is taking into account some of the context and, sorry, this is not, okay. And you can see that as the cell continues to evolve dynamically, that eventually this site is actually the one which undergoes fission. After this, the neural network does start to detect that one as being more probable to eventually undergo a fission. And so the idea that we had was to use this kind of event detection on the fly to toggle our microscope between a slow acquisition when it was unlikely that mitochondria would divide and a fast acquisition when the likelihood increased. And so here you can see the maximum event score. That's the integrated probability at an individual site that the mitochondrion will divide. And we set a sort of threshold range so that once the event score exceeded this upper threshold, then the microscope toggles to a fast imaging rate. So from 0.2 frames per second, it goes to 4 frames per second. And then once the score diminishes below a lower threshold after a fixed time, a minimum time, it switches back to a slow imaging rate. And so this is the basic concept for our initial implementation, that we would switch the rate of acquisition depending on the likelihood that an event of interest is taking place in the sample. And so with a slow frame rate, the fixed slow frame rate, we are able to capture mitochondrial divisions. This is already interesting and has been done. But with our event-driven acquisitions, we can actually switch on the fly, or the microscope switches on the fly, and you can see that during the event itself, we can capture a whole lot more data. And so this is interesting. This is useful. It's it gives us an enrichment of the data of the kind that we're interested in. So it's more or less what we've been going for, right? So the way that we think about this in terms of phototoxicity and the sample health and perturbation is that if we're imaging at a slow rate, we accumulate light dose at a fixed rate, but it's a slow fixed rate, but we're less likely to capture the events of interest. And if we capture at a fixed fast frame rate, then we accumulate light dose much more rapidly, photo damage much more rapidly. Now that we're switching between these slow and fast frame rates, then with event-driven acquisitions, all of these blue curves, you can see that we accumulate light dose at different rates depending on the individual experiment, but it's always going to be less than the fast and greater than the slow. Nevertheless, the idea is that during these fast accumulations, that's when an event is actually happening, and that's how we get enrichment of the events of interest. And so one of the measures that we had was, how narrow are the division sites that we're able to see? And you can see that the minimal width of the mitochondrion that we can capture is smaller in the case of event-driven acquisitions compared to slow acquisitions. We also were able to use this, repurpose this network to study bacterial division, mitochondria being ancient, having bacteria as their ancient ancestors. They actually undergo through similar shape transformations, and we were able to use the same network to enrich for bacterial cell divisions as well. And so the idea of this, again, this initial implementation is temporal adaptation between slow and fast imaging. Some of the advantages that this has is that you enrich the data sets for events of interest, you reduce the overall light dose, you increase temporal resolution. Of course, this kind of methodology is overkill for many kinds of biological problems. It's really best for imaging events that are rare and intermittent. That is, they're not happening all the time. So you gain value by imaging only when what you're interested in is happening. It's essential that there are some sorts of precursors that the neural network can pick up on to be able to do this on-the-fly switching. And of course, in any case, when you're interested in using AI or artificial neural networks, it's important that you've chosen your problem such that this is essential. And we do find that these kinds of events are too subtle to predict with simple algorithms like shape detection, intensity thresholding, and so on. And so things that we're working on now are, using this similar framework to detect different kinds of events, to do multicolor detection, and correlative between different imaging types, and as well as label-free detection, which I'll touch on a little bit toward the end of this talk. Yes, so we ran this LATSIS symposium on smart microscopy back in the fall, and this was a really fun opportunity for us to gather the community, which is really a nascent community. If you're interested in this, I would love to talk to you afterwards. I actually have to leave after this talk, but I am on campus, so don't hesitate to reach out. And one of the things that I came across when I was preparing for this symposium is this, I think, really beautiful example of how people have been thinking for a long time about the intelligent microscope. So I cited Jan Hiskin's work, but even before then, back in the 70s and 80s, there were people who were thinking about intelligent microscopes. And I love this work from Judith Pruitt. It's actually a published piece of work, Intelligent Microscopes, a Scientific Poem. And just to read a very tiny part of this really multi-stanza poem that I suggest you look up. So she cites this, quotes this work from Robert Hooke, who's one of the early developers of light microscopy, and she's adapted it to how she perceives the future power of the intelligent microscope. So with rapid contemporary advances in both scientific concepts and technology, it may be appropriate to modify Hooke's passage. It seems very probable that nature has very many appropriated instruments and contrivances, whereby to bring her designs and end to pass, which 'tis not improbable, but that some diligent observer, if helped with intelligent microscopes, may in time detect. So we're very excited about the possibilities that intelligent microscopes will eventually empower us to see things that we could not otherwise see. And indeed, so I showed you this data earlier, but perhaps many of you are not used to looking at mitochondria, but in fact, these event-driven acquisitions gave us a key insight into some part of the mitochondrial life cycle, which was previously obscure to us. And so this is related to, so I've mentioned the fact that mitochondria need to divide, to proliferate, and I've mentioned that they have their own DNA copies. Now, those DNA copies, they need to be distributed between daughter mitochondria for this life cycle to make sense, right? So every cell in our body has its own copies of our genome. Similarly, every organelle should have its own copy of the mitochondrial genome in order to perform its functions. And how does that work in mitochondria? They're lacking all of the machinery that our cells have to segregate DNA. And so in this movie, there are actually hints to what is segregating the mitochondrial genome. And so what you see are these membrane fluctuations which start to appear just prior to division. And these undulations occur at a specific length scale. And that length scale is set by the physics of the organelle. We're not the first to observe that this shape transition can occur. We are one of the first to remark that it can occur in this transient, reversible, and highly dynamic, rapid way. So this kind of instability is known as the Rayleigh plateau Rayleigh instability, or pearling. And so pearling because it looks like a series of pearls on a string. And this instability has been mentioned before in the physics literature, for example, where there's this, it's understood that there's this competition between the elastic energy of the system, the membrane elasticity multiplied by the membrane thickness, divided by, on the other hand, balanced by the membrane tension. So the elasticity tends to resist deformation, the membrane tension tends to round the organelle. And so the balance between the two tells you about this transition between a membrane tube and an undulating pearled tube. And so this gives you a dimensionless number called the capillary number. And depending on where the system is with respect to elasticity versus membrane tension, you can have either pearled states or tubular states. You switch between something that looks like a tube and something that looks like a pearl. And this is, you're familiar with this in your everyday life, because if you turn on your water faucet and then turn it off very gently, then eventually it'll go through this jetting to dripping transition. And it's also driven by these kinds of physics considerations. And so there's a characteristic length scale associated with this plateau Rayleigh instability, and that drives the relatively uniform shape, uniform size and distancing between these pearls. And when we look at them in mitochondria, we see that they're about half a micron in size, 483 plus or minus 86 nanometers. They're roughly spherical in shape. We observe that they take place over transient time. So between 5 and 15 seconds is the average time that we observe these pearled states living. And moreover, we have seen that mitochondrial physiology can be compartmentalized. So I'm not going to go too far into this, but mitochondria make ATP via a membrane potential gradient. And this allows the electrons to be driven through the electron transport chain. And so what we see here is that there are some parts of the mitochondrion which are working much harder to make ATP than others. So there, and this is occurring at the length scale of this pearling instability. We then could use the fact that we have these event-driven acquisitions. We could train a neural network to recognize mitochondrial pearls, and we could turn on then the channel to study the physiology just when that pearling transition takes place. And then we can see things now, for example, that the pearled part of this network is actually where the mitochondria loses its membrane potential. So there's an association between the membrane potential, that is the capacity of the mitochondria to make ATP, and the shape transition. What about DNA? So I mentioned that this is, oh, I actually don't include that here, huh? Well, if you're curious about seeing where the DNA goes, the DNA actually goes into the pearls. I don't know why I decided not to include that, but here I've mostly focused on the fact that you can use these event-driven acquisitions to capture this physiological data and do this switching between different fluorescence channels. So as a last part of my talk, I want to emphasize, trying to go even gentler. So I've been talking about switching between slow imaging and fast imaging, But in fact, there are other imaging modalities which are much gentler than fluorescence. And in particular, some of the oldest forms of microscopy, phase contrast microscopy, wide field microscopy, bright field microscopy, dark field microscopy, all use white light, broad spectrum light, and can go at much lower light doses to still capture shape information. And one of the things that we're lucky about is that, in fact, mitochondria show up very, very beautifully in phase contrast imaging. So here you can see the edge of a cell here. And in each of the cells, you can see some mitochondria as well as some other membrane structures. This, we can follow mitochondrial dynamics. And so you might think, great, you've already solved the problem of fluorescence. How much harder could it be to do the same thing in phase? And one of the issues with phase is that there's a lot else that is giving contrast to the imaging, and those give rise to false positives in our neural networks. And so, just looking at zooming into two of these structures, which is what the neural network would be seeing, you can see that it might be challenging to distinguish between structures that are associated with mitochondrial predivisions and with other features in the cell. And in fact, one of these is a mitochondrion and the other one is not. And so there are false positives that are detected, in particular, these edge of the cell ruffles that the neural network that we had initially trained really likes to see. They really feel like, this form looks very much like a mitochondrial predivision, but they're just membrane ruffles. And so one of the ways that we have adapted our detections, our whole detection framework is to take into account the dynamic signatures, because this is really what is differing between the two systems. So from frame to frame, in one case, you get this division and bouncing back of the two parts of the mitochondrion. And in the other case, this is maybe it's not playing. It is much less dynamic. These cell membrane ruffles, they last a long time. And so what we've been doing is to introduce time in different ways into our detection system. One way is that we are taking in multiple frames as inputs simultaneously as multiple channels. And we've looked at the influence of the number of time frames that we include as inputs simultaneously to the detection efficiency. We've also included a long short-term memory module at the bottleneck of the detection network. And this is what is really giving us a boost in performance. I'm not going into the details of that here, but we have a manuscript that will soon be on bioRxiv that describes the details of how we were finally able to solve this problem in a robust way. Yeah, so now we can capture these divisions more robustly in phase contrast microscopy. I think this has been a huge feat. This is a work of a fantastic postdoc in my group, Willie Stepp. And now we can turn on fluorescence just when mitochondria are about to divide to really reduce the light dose. The light dose is, in terms of phototoxicity, is near to zero during the phase contrast imaging. So this is really a high gain type of measurement. So here you can see in a bit more detail what happens as in phase contrast as the mitochondria constricts. Now the network sees it, identifies not only when, but where it's going to divide, and then turns on the fluorescence once the event score exceeds a certain threshold, images for a while until it divides. So this takes me to the end. Where are we going? I think smart microscopy has a lot of possibilities. We have focused in my group really on organelles and mitochondria. We're also looking now at mitochondrial contacts with other organelles, which we can see really beautifully in phase contrast. But we think that there's a lot of different types of biological events, which could be where this type of technology could drive discovery. Things that we're working on now, and actually people from my group in the room are working on spatial adaptation. So the neural network tells you not only when, but where. And so we would like to use sculpted light to be able to image specific parts of the sample and also potentially perturb them using optogenetic activation of those very precise locations at precise times. We also coming from a super resolution background, which is one of the most phototoxic forms of microscopy out there, we would love to be able to turn on a resolution switch, again, just to get down to the scales that are required to see what we would like to observe. And then we're moving into using other types of learning to identify outliers or other kinds of events of interest that perhaps we don't even realize are in there, using some different ideas with self-supervised learning. So our hopes are that eventually we're moving toward intelligence, and some people already use the word intelligent microscopy, but I think that there's quite a lot more that we would need to do to endow the microscope with more capacity to sculpt the measurement itself to investigate hypotheses, for example. But I'm very excited about the possibilities. Let me end by acknowledging some of the people in my lab who have led this work. Everyone in the group is involved in all projects, but the work that I talked about near the end was really spearheaded by Willie Stepp. Early on, Tatiana Kleil had painstakingly annotated many mitochondrial divisions and discovered patterns in mitochondrial division, and together with Dora Mahicic. And Dora had built our ISIM microscope and trained the initial neural networks to identify mitochondrial division, predivisions using the shape in fluorescence as well as DRP1. And then there's Juan Landoni, who's been instrumental in developing this purling project, identifying purling, and then also using it to study these different compartmentalizations that occur. And I'll thank our collaborators. Martin Weigert has been an amazing collaborator for all of the things related to artificial neural networks. He has taught us so much and helped us so much, in particular, devising different ways of incorporating dynamic information and the self-supervised work as well. And then our collaborators in this HFSP project grant who have continuously supported our work in mitochondria.